A very good evening to all of you. Today we will do microbiology challenge. It will be so. Let's begin with the first question. What is the preferred diagnostic method of HMPV infection? That is human metapneumovirus infection. A gram staining, B culture and chocolate agar, C reverse transcriptase PCR, D Vidal test. Well, Vidal test is used for Salmonella typhi. Culture on chocolate agar is used for bacteria. Gram staining is again used for bacteria. So the correct answer here is reverse transcriptase PCR. C. What is the natural reservoir of Marburg virus? This Marburg virus, A. Civet cat, B. Fruit bat, C. Rodents, D. Mosquitoes. What do you think is the correct answer? Please answer in the chat box, please. Please answer in the chat box. What is the correct option here? Natural reservoir for Marburg virus. Well, it is fruit bat, Rosettus egypticus, B. What is the natural reservoir for Marburg virus? It is a fruit bat, Rosettus egypticus. What is the average case fatality rate of Marburg hemorrhagic fever? It's quite high. Let us see the options. A 5 to 10%, B 10 to 20%, C 25 to 90%, D 95 to 100%. Well, the Marburg hemorrhagic fever has a very high case fatality rate. It is ranging from 25 to 90% depending on the outbreak and the quality of medical care. The case fatality rate underscores the importance of rapid containment and supportive therapy. So the correct answer here is C, 25 to 90%. Which population is most at risk for severe HMPV infection? Human metapneumovirus infection. A. Healthy young adults. B. Immunocompromised individuals. C. Adolescents. And D. Travelers. While HMPV can infect anyone, immunocompromised individuals, elderly patients, and young children are at higher risk of severe disease, including pneumonia and bronchiolitis. Correct answer here is B. Which of the following emerging viruses is primarily transmitted by the fecal oral route? A. Norovirus, B. Zika virus, C. Hendra virus, D. Rabies virus. This norovirus is an emerging virus that causes acute gastroenteritis and is primarily transmitted via the fecal-oral route, often through contaminated food or water. So the correct answer here is A. Norovirus. Draughtsman colony. Draughtsman colony means carom coin appearance. What is the shape here? Concentric rings are seen on the surface. When you view the colonies from above, colonies become flat with central flat with raised edges and central umbonation. So it is like carom coin, and it is seen in which organism? Shows this. A. Anthrax, B. Pneumococci, C. Pertussis, D. Yersinia. It is the pneumococci which shows this. Correct answer here is B. Which of the following viruses is not transmitted from animals to humans? A. Hendra virus, B. SARS-CoV-2 virus, C. Hepatitis C virus, D. Nipah virus. Hepatitis C virus is primarily transmitted through blood to blood contact, and it is not considered a zoonotic virus. In contrast, Hendra, Nipah, and SARS-CoV-2 have zoonotic origins. So the correct answer here is C, hepatitis C virus. Which of the following best describes the genetic material of SARS-CoV-2? Well, it is a single-stranded RNA positive sense virus. Single-stranded RNA positive sense virus. 20 patients got acute watery diarrhea on stool examination following motility pattern is seen. Culture media. Now look at the video here. What you can see here. Here you can see the darting motility is there. Darting motility is seen in Vibrio cholerae. Now let us look at the options. A DC agar, B TCBS agar, C McConkey agar, D blood agar. Well, it is TCBS agar. So the correct option here is B TCBS. Granulomatosis infantis septica is caused by A. Pseudomonas, B. Chlamydia trachomatis, C. Group D streptococci, and D. Listeria. Granulomatosis infantis septica is an intrauterine infection of newborn caused by Listeria. The predominant feature of this condition is extensive focal necrosis, affecting especially the liver and less often the lungs. The mortality rate is quite high. Occasionally, meningitis may be associated with it. The organism can be isolated from the affected areas in the child, and frequently it can be isolated from the genital tract of mother, who may or may not manifest the disease. So the correct answer here is D. Listeria. Stalactite growth is in key growth is due to the following organisms: A. Yersinia pestis, B. Treponema pallidum, C. Haemophilus influenzae, D. Corynebacterium diphtheria. The cultural characteristic of Yersinia pestis when it is grown in a flask of broth with oil or ghee floating on the top, a characteristic growth occurs which hangs down into the broth from the surface, resembling a stalactite. On nutrient agar, the colonies are small, delicate, transparent disc becoming opaque on continued incubation. On blood agar, colonies are dark brown due to absorption of hemin pigment. On McConkey agar, colorless colonies are formed. So the correct answer here is A. Yersinia pestis. Milk ring test is done to detect which organism present in milk: A. Bordetella, B. Brucellosis, C. Bartonella, D. Salmonella. Well, when the milk ring test comes, it's the brucellosis. For the detection of brucella in infected animals, pooled milk samples may be tested for bacilli by culture and for antibodies by several techniques. In the milk ring test, a sample of whole milk is mixed well with a drop of stained brucella antigen and incubated in a water bath at 70 degrees for 40 to 50 minutes. If antibodies are present in milk, the bacilli are agglutinated and rise with the cream to form a blue ring at the top, leaving the milk. So it is brucellosis. Superantigen causes A. Polyclonal activation of T cells, B. Stimulation of B cells, C. Enhancement of phagocytosis, D. Activation of complement. It is polyclonal activation of T cells. Respiratory syncytial virus causes fall: A. Coryza in kids, B. ARDS, C. Bronchitis, D. Common cold, except ARDS. Please click the link in the description box to join the 2-day Neet PG Breakthrough Blueprint Masterclass. The first 10 people who will to join the will get the masterclass absolutely free. Microorganisms which cause pyogenic osteomyelitis is A. Streptococcus, B. Staph aureus, C. Corynebacterium, D. Neisseria gonorrhoeae. It is Staph aureus which causes this pyogenic osteomyelitis. In nutrient agar, concentration of agar is A 1%, B 1.5%, C 3%, and D 4%. It is 1.5%. Which cell cause rosette formation with sheep RBCs? They are the T cells which cause rosette formation. Which is found in Dye syndrome? What happens in Dye syndrome? It is an example of T cell deficiency that result from failure of development of third and fourth pharyngeal pouches. The clinical features are enhanced susceptibility to viral, fungal, that is mucocutaneous candidiasis and bacterial infection, fecal facial abnormalities, hypertelorism, abnormal ears, short philtrum, and micronathia. Hypocalcemic tetany due to failure of parathyroid development, congenital heart disease of alert strategy, absence of normal thymus. Serum immunoglobulin concentration are frequently normal, but antibody response, particularly IgG and IgA subtype, are usually impaired. T cell levels are reduced, B cell levels are normal. So the correct answer here is A. Complement formed in liver are A C2, C4, B C3, C6, C9, C C5, C8, D C1. It is C3, C6, C9. Hereditary angioneuritic edema is due to A. Deficiency of C1 inhibitor, B. Deficiency of NADPH oxidase, C. Deficiency of MPO, D. Deficiency of properdin. It is due to deficiency of C1 inhibitor. Opsonization takes place through A, C3A, B, C3B, C, C5A, D, C5B. It is C3B. Staph aureus causes A. erythema, B. chancroid, C. acne vulgaris, D. bullous impetigo. Staph aureus causes bullous impetigo. So the correct option here is D. Which group of streptococcus grow at 60 degrees, that is high temperature? A, B, B, C, D. It is group D streptococci or enterococci. They are heat resistant and they can be grown at more than 60 degrees. Fish tank granuloma is seen in M. Fortusium, M. kansasi, M. marinum, M. leprosy. It is M. marinum. Fish tank granuloma, also called swimming pool granuloma, is caused by M. marinum. Aquarium granuloma, also known as fish tank granuloma or swimming pool granuloma, is a rare skin condition caused by non-tubercular mycobacterium known as Mycobacterium marinum. Skin infection with M. marinum in humans are relatively uncommon and are usually acquired from contact with contaminated swimming pools, aquarium, or sometimes infected fish. Botanist fever is caused by A. Rickettsia japonica, Rickettsia conori, Rickettsia zebrica, Rickettsia australis. Now, the tick-borne fevers caused by Rickettsia organism. Rickettsia conori, Mediterranean spotted fever, also called botanist fever. Kenya tick typhus, India tick typhus, South African tick typhus, Israel spotted fever, Astrakhan spotted fever, Rickettsia, Rocky Mountain spotted fever, Rickettsia Africa, African tick bite fever, Rickettsia japonica, Japanese spotted fever or Oriental spotted fever. Rickettsia honi, Flinders Island spotted fever. Rickettsia australis, Queensland tick typhus. So the correct answer here is Rickettsia conori. Which of the following is called praised knockout bacillus? Praised knockout bacillus is Corynebacterium pseudotuberculosis. Now, praised knockout bacillus, Corynebacterium pseudotuberculosis. Dark immortality, which is also found in, it is found in Campylobacter jejunum. XDR-TP is defined as resistance to fluoroquinolones plus INH plus rifampicin plus amikacin. This is the correct answer as D. Hebranos is caused by A. fish bacillus, B. staph aureus, C. pseudomonas, D. Corynebacterium bacteria. It is fish bacillus. Duodenal aspirate is used in the diagnosis of Giardia lamblia. Izumi fever is caused by Yersinia pseudotuberculosis. Far East scarlet-like fever is an infectious disease caused by gram-negative bacillus Yersinia pseudotuberculosis. In Japan, it is called Izumi fever. The signs and symptoms include red skin rash, usually on the face, elbow, and knees, skin desquamation, exanthema, red tongue, toxic shock syndrome. Other features include mesenteric lymphadenitis, arthritis, kidney failure rarely occurs. Relapses occur in 50% of cases. Unsegmented eggs are seen in which parasite? A. Tichuris tichura, B. Ankylostoma, C. Necator americanus, D. Didactylus. It is Tichuris tichura. Brazilian purpuric fever is caused by A. Bordetella pertussis, B. Haemophilus aegypticus, C. Haemophilus ducrei, and D. Haemophilus parainfluenzae. It is Haemophilus aegypticus. Brazilian purpuric fever is an illness of children caused by bacterium Haemophilus influenzae biogroup aegypticus, which is ultimately fatal due to sepsis.